know that some animals can glow?
How's that possible? I don't believe it.
Look! This is because they have a special type of bacteria inside them. This bacteria species is called Vibrio fischeri, and they produce light.
Hold on. Can I glow if I eat these bacteria?
Unfortunately, no. It only works in specific species. This is an example of bioluminescence, meaning the production of light by living organisms.
Wait, but if these bacteria exist, why isn't the entire ocean lit up?
The bacteria don't light up until there is enough of them together.
Why?
Imagine you were a bacterium. Why would you want to light up?
Oh yeah, why would I want to light up?
Well, who doesn't want to shine?
These bacteria shine because they want to be useful to the squid, so that they can be inside the squid and get nutrients from the squid.
Why is glowing useful to the squid though? Wouldn't it make the squid more visible to predators?
Nope, the opposite. Under the moonlight, where everything is lit up, the squid needs to glow as well to camouflage.
Okay.
Back to the scenario where you are a Vibrio fischeri bacterium. When is the best time for you to glow?
When I'm inside the squid.
Good, because that is when the squid needs the light. In the rest of occasions, you don't need to produce light because that would be a waste of energy.
How would I know when I'm inside the squid though, if I don't have eyes?
Great question. You use something called quorum sensing.
What?
This allows you to know how many bacteria of your own species are present around you. If there are a lot of you, this is likely because you are in a squid. But if there are not a lot of you, then you are probably just floating in the ocean.
But again, I don't have eyes, so...
I know. To detect that, you all secrete a chemical known as an autoinducer. If there is a lot of you, there will be a lot of the chemical. If the concentration of the chemical is high enough, the autoinducer will bind to receptors in the bacteria, causing the bacteria to light up.
I guess that makes sense.
What do you mean by receptors?
Receptors are proteins that can bind to chemicals and produce a response. We can call the chemical that binds to the receptor a ligand. In this case, the ligand is the autoinducer. You have a lot of these in your body.
What?
Yeah. You have receptors that bind to hormones, neurotransmitters, cytokines, calcium ions.
Can you go slower?
Of course. Hormones are signaling chemicals that travel in the bloodstream. Some examples you might have heard of include insulin, testosterone, and adrenaline, or epinephrine.
Oh, is adrenaline the fight-or-flight one?
Yes, we will come back to that later. Neurotransmitters are chemicals transmitted between synapses, meaning the gaps between neurons. Some examples you might have heard of include dopamine and oxytocin.
Is dopamine the one related to motivation?
Yep.
Campbell needs a lot of dopamine then.
Cytokines are proteins that are secreted to a very limited area. They usually have an effect on the cells that produced it or on nearby cells. Cytokines have a variety of functions, including causing inflammation. Lastly, we have calcium ions.
Calcium ions? Calcium as in the metal?
Yes, you heard right. They are used for signaling in muscles and neurons. You can see that different molecules travel different distances to send their messages. Neurotransmitters may only have to travel a few nanometers to reach the other neuron, but hormones may travel over a meter to reach its receptor.
That's a big difference.
Not all hormones and neurotransmitters are the same, though. Types of hormones include amines, peptides, and steroids. Amines are chemicals derived from amino acids. Some amine hormones you might have heard of include melatonin, thyroxine, and epinephrine. Peptides are chains made up of several amino acids. Some peptide hormones you may have heard of include insulin, glucagon, and ADH. And steroids are a type of lipid. Some examples include estradiol, progesterone, and testosterone.
The female and male hormones?
Basically, yeah. Similarly, neurotransmitters also have different types. We have amines, gases, amino acids, and esters.
What's the difference between amines and amino acids?
Amines are derived from amino acids, whilst amino acids are just amino acids.
And gases? Do we have gases in our brain?
Yep. Nitrous oxide is a neurotransmitter. You can have a look at examples of each type of neurotransmitter here. Now that we have talked about ligands, let's talk about receptors. As a recap, receptors are proteins that bind to ligands to respond to a signal.
How does binding magically make it respond to a signal?
Great question. Let's talk about signal transduction pathways.
What's that?
In simple terms, it is the process in which a signal is converted into a response. It is divided into reception, transduction, and response. During reception, the ligand binds to the receptor.
Is that all?
Yeah, that's reception. Here, the ligand is called the first messenger. The next step is transduction. Here, there can be two paths. If the receptor is a transmembrane receptor, meaning it's on the membrane, then the ligand binds to the receptor and the receptor catalyzes the production of a secondary messenger. The secondary messenger then catalyzes another reaction, which then results in another reaction, then another one.
Why would you make it so complicated?
This is because this type of ligand cannot enter the cell, so they need intermediates to relay the signal. This chain reaction is called a signaling cascade. If the receptor is an intracellular receptor, then the ligand binds directly to the receptor inside the cell, which forms a complex that can produce a response.
Hold on. Didn't you say that the ligand cannot enter the cell?
If the ligand binds to an intracellular receptor, they are a type of ligand that can enter the cell. I will expand on this in a bit. After transduction, you get the response, which is the end goal of the signaling. For example, producing more of a certain protein. Going back to your question of why some ligands can travel into the cell whilst others can't, this is because ligands have different chemical properties. Remember what were the types of ligands we had?
Oh yeah, we had neurotransmitters, hormones, cytokines,
And calcium.
calcium ions.
Good. And what about the subcategories?
For hormones, we had amines, lipids,
Steroids specifically, but yeah, they are a type of lipid.
and peptides.
Good. That will be enough to explain this. Some ligands, like amine and peptide hormones, are polar, whilst steroid hormones are non-polar.
And?
Let me explain. Membranes are mostly made of phospholipids, which have a polar head. This means that polar substances cannot pass through the membrane because they will be either stuck or repelled by the phospholipids. In contrast, non-polar substances can pass through with no problem. So, out of these, which ones will be able to pass through the membrane?
Amines and peptide hormones are polar, so they will not be able to pass through.
Good. This means that they can only bind to transmembrane receptors, which will then relay the signal.
And the steroid hormones are non-polar, so they will be able to pass through the membrane.
Great job! This means that steroid hormones can diffuse through the membrane and bind to intracellular receptors. Within these categories of receptors, we have even more subcategories.
Even more.
Yes! Within transmembrane receptors, we will be talking about gated channel proteins, G-protein coupled receptors, and tyrosine kinase receptors.
What's all of that?
I'll try to make it simple and fun. Plasma, Campbell, I will need you for this. Let's start with gated channel proteins. These are channel proteins that are normally closed, but if you give the right stimulus, they will open and let the molecule pass through. This stimulus can either be a ligand or a voltage that is high enough. An example of this channel can be found in your muscles.
What?
Yes, these are involved in muscle contraction. They are called acetylcholine receptors because the ligand is a neurotransmitter called acetylcholine. They are located on your muscles. When acetylcholine binds to the acetylcholine receptors, the channel opens, causing sodium ions to flow in. This causes a cascade of other events which makes your muscles contract.
Wow, that's interesting.
Next, let's talk about G-protein coupled receptors, or GPCRs.
What a long name!
Let's break it down. G-proteins are a type of protein that interacts with GTP, which is like ATP, but it's with a G instead. The G-protein coupled receptors are the receptors that bind to the G-proteins. This is how they look like in the inactive state. The GPCR is bound to the G-protein, which is bound to GDP, the form of GTP after it's split.
Oh, just like ADP and ATP?
Exactly. When a ligand binds to the GPCR, GDP detaches and GTP attaches to the G-protein. Then, the G-protein splits apart, and that can produce a cascade of events that produces a response. Let's give a specific example. An example of a hormone that uses GPCRs is epinephrine, also known as adrenaline.
Oh, the fight-or-flight one!
Plasma, you can be epinephrine, and Campbell, you can be adenylyl cyclase.
I'm what?
I'll explain later. Plasma, your job is to activate adenylyl cyclase, but you can only do so by binding to the GPCR.
Got it!
What did you do, Plasma? You broke it.
No, that's what's supposed to happen. This just means that the G-protein is activated, so the GTP bound to it splits. Now, it can activate adenylyl cyclase.
Oh.
Now you're activated.
Which means...
You can convert ATP into
ADP?
No, CAMP. This is a secondary messenger.
What was a secondary messenger again?
It is a molecule produced to relay the message from another message. In this case, it is relaying the response from epinephrine.
So, like this?
Yep, that's it. CAMP will do the job. It will undergo a series of reactions which result in glycogen breaking down to glucose phosphate. In other words, it will make the energy in your body available to use.
Oh, for fight or flight.
The last type of receptor is the tyrosine kinase receptor.
These names are so weird.
Not when you understand what it means. Do you know what tyrosine or kinase is? That's okay. Tyrosine is a type of amino acid, and kinase is a type of enzyme that adds phosphate groups to things.
How can these two things be related?
Tyrosine kinases phosphorylate tyrosines. Let's dive into an example straight away. Insulin uses tyrosine kinase receptors.
Is insulin the one that makes cells take up glucose?
Yes, it is. When insulin binds to the receptor, these two parts join together. Then, the tyrosine kinases phosphorylate each other. This activates a series of reactions which results in a response. One of the responses is that it moves vesicles with glucose transporters to the cell membrane. So the vesicles fuse with the membrane and the transporters become embedded in the membrane. As the name indicates, glucose transporters transport glucose into the cell.
Oh, wait, so this is how insulin makes cells take up glucose.
Gated channels, GPCRs, and tyrosine kinase receptors are all transmembrane receptors. But what about intracellular receptors?
Oh no, are you gonna talk about a bunch of other types of intracellular receptors?
Nah, I'm just gonna talk about them in general. Since intracellular receptors are inside the cell, only non-polar molecules like steroid hormones can cross the membrane to bind to them. Once they bind, they form a complex that can change gene transcription. Gene transcription is the start of protein synthesis, so this changes how much of a particular protein is produced. This has an effect on the cell. The end.
That's it.
Yeah, that's it. Some examples of steroid hormones include estradiol and progesterone. These are produced by ovaries. When they are released, they bind to intracellular receptors, forming complexes that influence gene transcription. The response they produce is different though.
How?
For progesterone, when it flows into endometrium cells in the uterus, it increases transcription for the gene that codes for the insulin-like growth factor.
So what?
This makes the endometrium grow. The other hormone I'm gonna talk about is estradiol. At low levels, estradiol inhibits the production of a hormone called gonadotropin-releasing hormone, or GnRH.
Another long name.
It's basically a hormone involved in the menstrual cycle. You will learn about it in detail at some other point. What is interesting about this is that GnRH leads to the production of estradiol, which means that when the levels of GnRH drops, the levels of estradiol also drops, but that means that GnRH isn't inhibited anymore, so its levels increase, which means that the levels of estradiol also increase, which means that GnRH...
Is the cycle just gonna go on and on?
Yeah. This is also known as negative feedback, where the result of a pathway inhibits the initial signal, therefore controlling the amounts of both within a range. What is even more interesting is that when there are really high levels of estradiol, the story changes. Now estradiol will promote the production of GnRH. This means that when there is estradiol produced, there will be more GnRH produced, which means that there will be more estradiol produced, which means...
Okay, we get it.
This is called positive feedback.
It has to stop at some point though, right? Or otherwise the body is just gonna fill up with estradiol and GnRH.
You're right. GnRH also leads to the production of progesterone, which inhibits estradiol, so the levels of estradiol eventually drop.
Can chemical signaling make me glow?
Only if you're a squid.